Oh, I didn't open up my PowerPoint. Today was one of those Mondays where I woke up and was like, oh, it's a nice Sunday. It's, no. Okay, now we should be going. All right, so we started the digestive system last week, just talking about the basic structures. Today we're going to talk about the general functions of each of these organs in a little bit more detail than we went through on Friday. And then Wednesday we're going to bring in the regulation, so how are each of those systems being regulated from the nervous system, from chemoreceptors, and all those fun things. And then as a reminder, this Friday, there is a lecture. It's digestive ancillary function, so the function of like the liver and the pancreas and all that fun stuff. But I won't be here, so I'm going to be recording that lecture. I'll post it by Friday morning at the latest, so you guys do not need to attend my class on Friday. The lecture will just be posted for you. All right. So we will follow the same kind of pathway that we discussed on Friday. We'll start at the mouth and work our way down. So we start the functioning of your digestive tract in the mouth with the process of chewing. So remember, chewing is the mechanical or physical breakdown component of making that food that you're consuming smaller, more usable pieces that will then be further broken down later on in the system. It's going to do so through a variety of movements. You can call it mashing, slicing, pulping. There's all sorts of different terms that are applied to the various ways in which you can move your teeth in relation to each other. And the goal is to create smaller food that is going to be easier to digest by your stomach and the acids inside of it. You are also doing some form of chemical breakdown by introducing saliva, which has digestive enzymes in there. This saliva is going to provide moisture to your food to turn it into a slurry. And then again, further on, we're going to add even more moisture. We're going to turn it into essentially a liquid moving through your digestive tract. The chewing portion is done by your teeth. And as I mentioned on Friday, there are different types of teeth that are going to be doing different types of chewing. So you can break it down into really two regions of the mouth. The anterior portion, so right in the front and then a little bit off to the sides, you have your incisors and your canines. These are going to be more shovel-shaped teeth. They don't have a large flat surface. They have kind of a long ridge and they're scooped on one side. And the motions that these teeth are responsible for predominantly are slicing, snipping, scooping motions. These are the types of teeth you associate with animals that consume meat, things that are tougher that need to be cut through. Premolars and molars, these are going to have those nice flat surfaces, and they're going to be predominantly doing things like grinding and mashing and pulping your food. So you can break these down into these two primary categories. Now, these teeth can do really any of these motions. Your incisors can do some grinding and some mashing. They're just not nearly as efficient at doing that as the back of your teeth or as your molars. So I'm sure if you've ever stopped to think about your motions of your mouth while you're chewing, you can absolutely do all these things anywhere in your mouth, but it's going to be easier to, for example, bite through something tough using your front teeth than trying to just rip it off with your molars. Our teeth are what we call occlusal teeth. These are teeth that are designed such that the upper teeth and the lower teeth are going to fit together nicely. They meet up so that there's very little gap between your upper and lower teeth. This is called occlusion. There's a number of different ways in which teeth may not occlude properly. So you can have things like overcrowding. I have a little bit of overcrowding in my mouth. You can have an overbite where the upper teeth stick out too far forward, and so you don't have the nice occlusion at the front, making slicing motions much more difficult. And you can have an underbite where the underside sticks out forward, and again, you're going to not have that nice occlusion happening. These are all going to, in some way, affect your ability to chew and pulp your food. Most of these can be fixed through dental care in some form. It just depends on the degree to which that misalignment is occurring. For example, like I said, I have some overcrowding in my teeth. My dentists have all said that it's not to a point where I need to actually physically change it. It's just causing a minor misalignment in the very front. So for not everyone needs to. Some people absolutely need to have realignment happen. This process, or this method of occlusion, is something that is common amongst mammals. So you have it, but other mammals have it as well. And it's the reason that mammals only have 2 sets of teeth. So we have those baby teeth that fall out when you start to eat more dense, tougher foods. And you get those adult teeth that come in. And the proper method would be those adult teeth grow in and they occlude properly, and you can grind your food and you'll survive just fine. Other animals, like reptiles, for example, are constantly losing and replacing those teeth. Now, if you're constantly losing and replacing your teeth, you are running the risk of those teeth not occluding properly when the new ones come in. So we've lost that trait. Mammals just grow that adult set of teeth, hopefully they occlude properly, and we're not going to risk making it come in misaligned another time. So we just get those 2 sets of teeth so that they stay occluded properly. All right, so inside the mouth, we have a structure we call the palate, and that's this structure right here. This is the structure that separates our nasal cavity from our oral cavity. And it is 2 parts. There is the hard palate, which is the bony region of the roof of your mouth. And then there's the soft palate, which is a region of just tissue that hangs off the back of the hard palate. So if you were to explore the roof of your mouth with your tongue or a finger, you'll feel that it's very hard up front. That's because there's bone immediately on the other side of that tissue. But if you move further back, you find a ridge where suddenly it's not solid anymore. That's the boundary of the hard palate intersecting with the soft palate. And the soft palate is moving all the time. It's part of how we make sounds. And so that one is not rigid. It flexes and it moves all the time depending on what it is you're doing. The evolutionary reason we have a palate is that it allows us to eat and breathe at the same time. It has created 2 separate channels, our nasal cavity and our oral cavity, so that we can be chewing food and still be taking in air. We can breathe through our nose while we're chewing. Now, this excludes swallowing. You cannot swallow and breathe at the same time because you are actively closing off the trachea during the swallowing motion. But during chewing, you can do both. There are many different organisms that can't quite achieve this, although it's less of an issue in modern animals. I'm thinking more fossil organisms. There's quite a few that had a connected cavity, and so we don't anticipate that they could chew and breathe at the same time. All right, the process of swallowing. I have brought this up a couple of times, so now I'm finally going to talk about it. Food, which is often referred to as bolus, that is the term for a mass of food that you are in preparation of swallowing, is going to be pushed backwards by your tongue. So your tongue lifts up and pushes that bolus towards the back of the mouth. The soft palate, remember that is a fully soft tissue and it is movable, is going to move back and downwards to close off the nasal cavity. So this is going to prevent that bolus from going up into your nasal cavity, because that would be bad. That's the first 2 steps. Now here is where I need to point something out, because if you follow along with your book or use your book for studying, the book has an error here. Your book says the next step is the epiglottis, which is this rigid kind of cartilaginous structure that sits right here at the top of your throat, is going to be pushed downwards and cover up your trachea, and that's how you close it. That is not what happens. It's a nice, efficient way. It'd probably be more efficient than what we actually do, but that is not how it functions. So just to be clear, if you're studying your book, that is an error. What actually happens is your larynx, so right here, that top of your trachea, is going to physically move upwards, and it runs into the epiglottis, which is mostly immobile, and it acts like a trash can lid. So your larynx lifts up, hits the epiglottis, and is closed off. So food is not going to go down into your trachea. So, and you can see that happening in this video here if you watch him. Here's his larynx right here. He swallows, and it physically moves up. Same thing if you want to. You can put your hands right here and swallow. You will feel your larynx shoot up in your throat. So just to make sure you're all clear, that's the way it actually happens. And this is going to ensure that food does not go down the trachea. Instead, it's being directed backwards into the esophagus so we can head down into the digestive system. So now we need to move that bolus from our pharynx down through the esophagus. And remember, the esophagus is really just a tube. It's not doing any form of digestion for us. It's just moving that food material from your mouth down to your stomach. It is going to be entirely made of smooth muscle, and that smooth muscle is going to contract in such a way that it will push that bolus down the tube towards the stomach. So it's going to contract immediately behind the food and relax in front of it so that you squeeze and push that food forward in the tube. This process is called peristalsis. So the physical process of your esophagus squeezing behind a food particle and pushing it farther into the system is peristalsis. And you can kind of think of it as a wave of contraction. So it's not just contract here and then we'll contract a little bit further down and a little bit further down. It's like a wave. It starts and it just propagates down the entire esophagus. You may recall that the esophagus is quite a bit longer than you might initially anticipate. So it starts here at the top of your neck, and it's going to go all the way down through the abdomen, pass through the pass through the thoracic cavity, through the diaphragm, into the abdomen, where it meets up with the stomach. And each wave of peristalsis is going to take approximately 10 seconds to travel all the way down the esophagus. So each time you put food into your esophagus, each wave of contraction to move that food is taking approximately 10 seconds. Then we enter the stomach. Remember, we have to pass through that cardiac sphincter. That is the first entry point. And the stomach is then going to use additional waves of contraction, now not to move the food, but to mix the food. So remember, I talked about this on Friday. There are 3 layers of muscle tissue in the stomach, and each one of those layers is oriented at a different direction, so that as they contract separately, they are grinding or pulping or mixing the food in a churning motion. So it's lots of different directions of being contracted at the same time. It's also introducing fluids in the form of acids. So this is your stomach acid, and that is going to further help chemically break down your food to turn it into a slurry, what is almost entirely a liquid at this point. So once your stomach has gone through this series of contractions and it's turned your food, it's mixed it into acid, it's going to turn it into a mostly fluid material, it's going to start releasing small quantities of that material in through the pyloric sphincter into the duodenum. So you want to make sure you don't think of it as like a flushing motion. It's not everything all at once leaves the stomach once it's been slurried. It's going to release small amounts at a time. So your pyloric sphincter sits right here. It will receive the proper signal telling it to open up just a little bit, let a small amount of that material through, and start sending that through the duodenum. The material being pushed through is mostly fluid, and we give it a new name at this point. So after it has been churned by the stomach and mixed with acid, we call this chyme. This is not to be confused with chyle, which is the material I talked about with the lymph system, which is lymphatic fluid mixed with fats. That is a completely different thing, but it sounds very similar. So we can have a quick discussion of health concerns associated with this particular portion of the digestive system. You can have something called acid reflux. And this is where a stomach is going through that churning motion. It's adding acid to the food, and the cardiac sphincter, the one at the top where the esophagus attaches, is not fully closing. And so during this churning motion, it can slosh acid back up into your esophagus. Now, your esophagus has no anatomical defense against acid. It's not meant to have to survive acid moving through the esophagus. And so it actually starts to burn and degrade the tissues of your esophagus. And that's why heartburn, which is the common term for acid reflux, is painful. You feel a slight or intense, depending on the occasion, burning sensation along this esophageal tract. This is one of those things where some people will have it on occasion. I'm one of those on occasion. After eating certain foods, I am prone to having heartburn. I can usually deal with that in a case-by-case basis. Some people have chronic heartburn or chronic acid reflux, and if that is not taken care of medically, you can actually burn through your esophageal tissue and cause permanent damage. So some of the things you can do to take care of heartburn: you can do common treatments like taking calcium carbonate. That is what Tums are. If you've ever taken Tums, it's literally just calcium carbonate. And that is a base. So when you mix a base and an acid, you are neutralizing that acid. And so you neutralize the material in your esophagus and stop that burning sensation. Now again, if this is a chronic condition or something that you experience very regularly, there are other treatments that involve different medications you can take, either prescribed or over the counter, that actually block the acid production system in your stomach. We'll talk about that system on Wednesday, but that system can be blocked so that it's not telling it to make more acid, and it leaves your system with just enough acid to turn your food into chyme. It doesn't produce excess to try and minimize the amount that could be sloshed back up into your esophagus. All right, heading into the duodenum now. The chyme is going to have digestive enzymes from the pancreas added to it. This is going to help further break down that food chemically. We are also adding bile from the liver and the gallbladder, and that's going to help with the breakdown of fats. And these digestive enzymes and bile are going to be added. You can just barely see it. There are two small openings here on the lining of the duodenum. These are called the major and minor duodenal papillae. So they're just ducts, they're just openings, and they line up with the ducts of the pancreas and the ducts of the gallbladder and liver to add these materials to the chyme. This is then going to progress into the small intestine. One other thing that does happen here, the pancreas also adds bases. So just like heartburn is that acid getting up into the esophagus where it's not meant to combat that, your small intestine is not able to combat acids either. But your body has a mechanism to prepare for that. It adds its own bases to your stomach acids at the point, same thing through the major duodenal papilla. And that's going to neutralize the acid in the chyme. And so by the time it actually enters into the small intestine, it's no longer acidic. So this is like a natural form of taking Tums. moving the material through the small intestine is going to be similar to what happens to the esophagus. But now, rather than those contractions just moving it forward through the tube, there's going to be a series of contractions that are actually moving the food back and forth within that tube. So we call this process segmentation. If we look at this figure here, we can say these are two unmixed pieces of chyme. It squeezes here. We make little balls on either side. And then it's like you shift that squeezing to where the balls are, and you just squeeze it back and forth, back and forth. I always think of it, and maybe this is showing my age, but I had a toy as a child. This was like a little water tube, and you could squeeze it, and it would go back and forth, and there was usually glitter or something in there. That's the way I think of it. And it's just moving that material back and forth in your tube in order to mix all of that chyme and make sure that it is a uniform consistency and uniformly mixed with those digestive enzymes, because the goal is to make it as efficiently digestible as possible. So again, this figure is showing you undigested, or unmixed, I should say, partially mixed, and then by the time you have finished going through the small intestine, it is fully mixed, and we are starting to remove those nutrients. This also slows the food movement because we're not just squeezing it through the tube, we're making it take its time, and that's going to increase the amount of time that the small intestine has in contact with that food to ensure an efficient exchange of nutrients. Now, the small intestine, I talked about the gut tube in general on Friday with the various tissue layers. The interior layer is called the mucosa, and it is heavily, heavily folded. It is folds on folds, and this is going to increase the surface area on the inside of that lumen, inside the small intestines. If we look at this figure here, you can see that the wall is folded, and then the wall of those folds is folded again. This is making sure that you have the optimal amount of surface area for exchange, making sure that this exchange of nutrients is going to be as efficient as possible. Okay. Here is a tissue slide. We're going to be looking at this not this week, but after spring break. We'll have these in lab, at least in my class. And so you can see here the larger scale folds on the wall of the small intestine. And then within those, you still have all these little finger folds on the surface of those larger scale folds. So lots and lots of surface area. So let's talk a little bit about those folds. Those tiny folds that are sitting on the larger scale folds have their own name. We call these villi. Singular would be villus. And they are covered in absorptive cells. Those are the cells that are going to be absorbing the nutrients, absorbing vitamins, to take that in and send it into the bloodstream. To that end, each of these little finger-like villi are full of vasculature. So you have little capillaries coming up into that tube and little veins leaving it so that we can exchange both the material that the cells need to survive, but also take all of those nutrients that are being absorbed and send that into the bloodstream to be processed somewhere else in the body. So make sure that you get this concept in your head that these are 3D little finger-like projections on the inside of your small intestine, because we're going to see the exact opposite structure in a different structure a little bit further on. So what's happening with these little villi? We have biomolecules and nutrients that are being absorbed by those cells. All of that's going to be sent into that vasculature, sent into the bloodstream. But along that series of absorptive cells, there are also a few other types of cells. The only one I really want you to be aware of are these cells we call goblet cells. They're shown in these yellow kind of oblong structures in this figure here. And these are going to be spaced out along the edges of the villi, and their job is to secrete mucus. Just a thin layer. It's not an excessive amount of mucus. It's just a layer to keep the surface of these absorptive cells protected from anything that might be passing over it that could be damaging, as well as to make them sticky. This is part of how they're going to grab onto food particles and absorb the nutrients from them. So again, if we look at this figure, you have absorptive cells. They're typically much more rectangular in structure. Again, we'll look at this in more detail in a lab. But we also have these goblet cells, which tend to be more rounded, or I like to think of them as kind of like beaker shaped. And those are going to be producing the mucus. Okay. So the small intestine takes whatever water and nutrients it's absorbed. It's going to send it into the bloodstream. Now, these absorptive cells aren't super picky. They don't have their own mechanism for filtering anything. They just take whatever they can absorb from the food and put that into the vasculature, into the bloodstream. Therefore, all of the blood that is coming out of the intestines is first going to go to the liver. And the liver is going to detoxify that blood, take out any of those pathogens or harmful materials, so that whatever blood it passes back into the circulatory system is only full of the good nutrients, the things that the body does need. And it's going to do so through that hepatic portal system that I mentioned when we talked way back about vasculature. So if we look at our figure, we have these very heavily branched vasculature for the intestines, because you have a lot of surface area there. You need to take in a lot of blood. All of that blood, all of that vein structure is going to head to the liver first through this thicker vein we call the hepatic portal vein. It's going to pass through the liver, detoxify that blood, take out any of the pathogens, any of the toxins that are in there, and then send it back out through the hepatic vein into the inferior vena cava, back into the rest of the circulatory system. So all blood from the digestive system has to go through the liver first to make sure it's nice and clean, and then everything else will go back into the system. Now, when I talked about the thoracic cavity, you learned about a couple of different sacs that your organs sit in. For example, the heart sits in the pericardial sac, and the lungs sit in the pleura, which are just the equivalent, but for the lungs. We have the same thing for all of your abdominal organs. Instead of one individual for each organ, instead we have just one large sac, and it's called the peritoneum. And it's exactly the same as those other two sacs. It is like a balloon that you've pushed a structure into, and it's enveloped it on either side. And so just like that water balloon analogy that I've used before, you have a double membrane surrounding your organs, and that double membrane is full of fluid. It's going to be doing the same jobs as it did for the heart and lungs. It's going to provide a cushion, it's going to provide protection and space for extension. Again, your stomach changes size as it goes, and so you need a little bit of room for expansion there. It also serves an extra purpose, and that is suspension. You don't really think about it, but your abdominal organs are kind of just hanging in your abdomen, and they're sitting on top of each other. And you don't want to have a lot of friction between each of those organs. And so you have this balloon or water-filled balloon structure that kind of separates them and suspends them. It allows them to be mildly buoyant within your stomach, or excuse me, abdomen. Stomach is an organ. Now, that peritoneum is going to cause a little bit of difference compared to the the pleura and the pericardium, because we have multiple organs sitting in the abdomen inside this peritoneum. We need to have a way to get blood to all of those organs. Again, all of our nutrients is coming through that system, so we need to have an efficient system for that. So these blood vessels need to get through that peritoneum in order to reach each of the digestive organs. It's going to do so through these structures we call mesenteries. This is a tunnel or tube, you can think of it, that's pushed through the peritoneum full of vasculature to reach the organs that are inside. So if we look at this figure, here's our abdominal organ sitting in its peritoneum. We're going to push a little pathway to that organ, and that's how the vasculature or the nerves are going to get to that organ. And it creates a fold, a thin layer that essentially follows the entire ridge of every organ. So if you look at this anatomical picture here, this is a picture of small intestine that's been lifted up. And this is a very thin layering of that peritoneum, and the blood vessels are all pushing through that to reach the side of the small intestine. So it's just a fold in the peritoneum that allows the blood vasculature to reach into the peritoneum to reach the organs. All right. We talked about Peyer's patches briefly when we talked about the lymphatic system. Gosh, that's a couple of weeks ago now. These are specialized lymphatic structures, and they are located in the small intestine, and specifically in the distal ileum. So remember, your small intestine is separated into two halves, the jejunum at the beginning and the ileum at the end. And this is at the very end of the ileum. So this is mostly going to be concentrated right before it passes into the large intestine. These are going to be small cell capsules that are going to be inhabiting the mucosa layer, so the most internal layer of your gut tube. And that means that they are exposed to the lumen so that they can do their job. They're going to be monitoring for pathogens, just like any of our other lymphatic structures. It's just that it's monitoring that chyme that's passing through the digestive tract rather than monitoring lymph, like the lymph nodes, or blood, like the spleen. It's just monitoring chyme now. It's looking for pathogens, any type of antigen that's not recognized by the body. And just like the lymphatic system, it's going to react in the same way. It houses their own lymphocytes, so it has its own macrophages, its own T cells, its own B cells. So if a pathogen is detected, we're going to use that adaptive immune response. The B cell is going to eat a pathogen, make its own antigens. It's going to meet up with a helper T cell. It's going to start making its antibodies, that whole two-step verification process that we learned about. So it's the exact same function, it's just in the small intestine now rather than within the lymph system. All right, large intestine. This is going to be doing something similar to the small intestine. We're still going to have that contraction happening, but it's going to be happening much less than in the small intestine. So the segmentation, that squeezing back and forth, is going to be much more minimal. And mostly, the large intestine is going to be trying to move that material forward in the system. There's not as much back and forth material. Mostly it's forward. It's also much rarer. So the squeezing, the contraction happening in your large intestine only happens about 3 to 4 times a day. So rather than a constant back and forth like the small intestine, you're going to have these, what we call mass movements, where there's going to be a large contraction and moving that material forward in the tube. Typically, these are going to be triggered by having consumed food. When you eat, your body receives the signal that we need to make room in the digestive tract, and so you'll have a mass movement, squeeze some of that material forward and make room behind it for any incoming food. Some of the triggers of that would be distension, so stretching of the stomach, as well as in the duodenum. The duodenum can sense that chyme is being added into that tract, and that will trigger these mass movements. Now, the small intestine is doing a mass majority of the absorption, especially nutrients. However, the large intestine is going to be a lot of water absorption. So we kept water mixed in with that chyme all the way up to the large intestine, because that allows that material to move nice and easy through the stomach, through the duodenum, through the small intestine. But now your body wants to retain that water. We need to keep water to keep ourselves healthy, so we don't want to just pass all of that out of our system constantly. So your large intestine is primarily functioning to absorb that material, to absorb that water to keep it within your system and then just expel the hardened, compacted food remains. There is also some vitamin and mineral absorption happening in the large intestines, particularly vitamin K. Vitamin K is a vitamin that is essential for blood clotting as well as bone building, and it's actually something that we don't get much from our diets. Our primary source of vitamin K is through gut bacterial production in your small intestines. So your water absorption is going to be aided by these structures we call colonic crypts, also sometimes called intestinal glands. I like colonic crypts, sounds a little bit more interesting. Now this is where I said these are going to be kind of the opposite structure of those villi in your small intestines. So these are going to be small tubes, but rather than sticking out into the lumen, they're like pushed into this structure of the gut tube. So they're backwards. So if we look at this figure, rather than being open to the lumen, they get pushed back into the tissue of the large intestine. And they're going to be doing a vast majority of the water absorption. And they do so by using cells next to the lumen that are producing sodium. They're pumping sodium into the water sitting inside those tubes. And what this does is increase solute concentration so that osmosis kicks in and the water traveling through the lumen of the large intestine gets pushed into those little tubes to try and dilute that solute. It's that natural passive movement of fluid, simply because we've increased a solute concentration within each of those crypts. This is then going to suck the water out of the food that's traveling through the large intestine to make sure that we keep it for the function of the body. As I mentioned, we have our own gut flora. These are intestinal bacteria that are supposed to be there. We want these bacteria. They help break down our food, and they provide us with essential vitamins. So this diverse ecosystem of bacteria are going to be living off the remains of the food passing through your digestive system. So they're doing kind of a symbiotic relationship here. They're getting their nutrients from your food, and in return, they produce essential vitamins like vitamin K. And again, like I said, that's necessary for blood clotting and some parts of bone building. And this is the primary source of vitamin K. We don't eat much of it through our diet, so you need a healthy gut flora in order to get this vitamin K. So to summarize large intestine, we've got that food coming through. At this point, it's mostly liquid, that form of chyme. Most of the nutrients have already been removed by the small intestine, and now the goal of the large intestine is mostly to absorb water and then some of that vitamins. Part of the way in which the large intestine does this is it's going to squeeze during those mass movements, and it forces the water component of the chyme forward faster than it forces the compact food remains through the large intestine. This is going to result in dehydrating the food remains and making sure that the water is nice and easy to access by those crypts. So there's a little animation I have here. This is our chyme entering the large intestine. It's a mix of water and food. As we move forward, we separate those from a mass movement. The yellow structure, that's going to be the food components. The blue is the water, so we've squeezed and we're forcing the fluid component out of the chyme a little bit faster. We go through another mass movement and that separation is going to become even more obvious. So now the food is back here It's being dehydrated. We've squeezed the water out of it, and now those contractions are just going to be compacting it, making it smaller and denser. The water, on the other hand, is now passing through our large intestine on its own. It's heading into those crypts where it can be absorbed through osmosis. And as that food passes through, it's getting dehydrated, compacted, and that water is getting smaller because we're absorbing it through that last portion of the large intestine. Last stop. So that compacted, dehydrated food waste is what we call feces. And this is going to be all the undigestible foods, things like cellulose, some fibers, things like that, things that we can't get nutrients out of. And then also, you may or may not know this, feces is predominantly made up of bacteria. I was looking this morning, it's somewhere between 50 and 80% depending on your diet. So a vast majority of the feces is bacteria being passed out of your system, mixed in with that undigested food. So once we have created feces, and feces has made it all the way through the large intestine, through the sigmoid colon, it enters into the rectum. Having entered into the rectum, it causes that tissue to stretch. That's what distension means. It's just stretching. And that stretching is going to trigger your series of sphincters to react. You have an inner sphincter and an external sphincter. These are going to be surrounding the anus. The inner or internal sphincter is immediately adjacent to the walls of the anus. The external is a little bit further out, so it's not like there's one stacked on top of each other. They're right next to each other. One's just closer to the internal structure of the anus, one's a little bit further out. The inner sphincter opens automatically. It's completely unconsciously controlled. It is triggered by that stretching. So once feces has moved into the rectum, has caused it to stretch, the inner sphincter opens because it says, okay, we need to make room, we're going to pass this out, we're ready for defecation. The external sphincter, on the other hand, is what you as toddlers learn to control consciously and get to decide whether or not that gets opened. And that's how we can control our urges to go to the bathroom. So that particular sphincter will not open without a person's consent unless it's become an emergency situation. All right, key topics, the mechanisms for swallowing, make sure you get that clear. Movement in the gut tube at each of the stages, how is it being moved? Is it peristalsis? Is it mass movement? That sort of thing. The jobs of each organ, if it's adding anything, if it's absorbing anything, and then what is that mesentery structure? Because I think that's a little bit more of a confusing thing to visualize. Wednesday, digestive regulation, and then as a reminder, Friday's lecture will be recorded and posted online. You do not need to attend on Friday.